The water is motionless when the line breaks. A silver ribbon suspended in the midwater column appears inert, aligned with the current, as if it were debris polished by sun. A school of sardines drifts along the reef edge, maintaining loose formation, each fish reading the minute pressure changes of its neighbors through the lateral line. There is no sudden shadow, no eruption from below, no swirl of sand to betray danger. Then the silver line folds into velocity. The barracuda's body contracts and extends in a single decisive arc, and the distance that seemed safe collapses to nothing. One sardine disappears into a mouth lined with daggered teeth, and the rest scatter into fractured geometry. The ocean returns to calm, but the stillness has been redefined as prelude. The barracuda is an embodiment of latent force. Its elongated, torpedo-shaped body is built around axial musculature, optimized for rapid acceleration rather than sustained cruising. White muscle fibers dominate its posterior flanks, capable of generating explosive contractions that propel it forward in a straight, uncompromising line. Unlike predators that circle or herd, the barracuda often commits to linear interception, trusting precision over spectacle. The body's narrow profile reduces drag, and its deeply forked caudal fin translates stored muscular energy into sudden displacement of water. In biomechanical terms, it is a projectile with sensory guidance. What makes the barracuda's strategy unsettling is its relationship with stillness. It often hovers almost motionless in open water, head slightly elevated, pectoral fins adjusting subtly to maintain position. In sunlit midwater, where particulate matter glitters and currents shimmer, the barracuda's reflective scales mirror the environment, fragmenting its outline. This mirror-like quality is not mere aesthetics. It is camouflage through optical confusion. By reflecting the surrounding blue and green hues, the fish dissolves into the column of water itself. The predator is visible yet unreadable, present yet not immediately categorized as threat. The first narrative reversal emerges here. In ecosystems where speed is equated with danger, stillness becomes misinterpreted as safety. Prey species are tuned to react to sudden motion or looming shadows, neural circuits primed to detect acceleration rather than quiet alignment. The barracuda exploits this bias. It does not conceal itself completely. It presents itself as neutral. The pause before movement is not hesitation, but calculation. Within that pause lies the compression of energy, the moment when muscle fibers prime for contraction and sensory systems finalize trajectory. Vision is central to this precision. Barracudas possess large eyes adapted for high-resolution detection in well-lit coastal waters. Their retinas contain a high density of photoreceptors, allowing them to resolve fine details and track individual prey within schools. In the refracted light near reef edges, where clarity can fluctuate with tide and plankton density, this visual acuity enables targeting of specific individuals. Rather than scattering energy across the entire school, the barracuda selects a single fish, aligning its body with predicted movement. The strike is not random lunge but calculated intercept, guided by milliseconds of sensory processing. Electroreception and lateral line sensitivity further refine this targeting. While not as specialized in electroreception as some sharks, barracudas detect pressure waves and minute disturbances in water flow, reading the collective movements of schooling fish. A slight deviation in alignment, a fraction of hesitation within the school, can signal vulnerability. The predator's neural circuitry translates these cues into motor output with minimal delay. The silence before speed is neurological efficiency at work. The reef edge provides context for this ambush style. Here, open water meets structural complexity, creating corridors where schooling fish transit between feeding and shelter zones. These corridors are rich in prey density, but often lack the tight crevices that favor ambush specialists like morays. The barracuda occupies this intermediate space, where neither full concealment nor complete openness defines the terrain. It becomes a sentinel at the boundary, its metallic body suspended like punctuation in blue syntax. The second narrative reversal unfolds when considering vulnerability. The barracuda's reliance on explosive acceleration demands high energy expenditure, and repeated failed strikes can deplete reserves. Its elongated body, while efficient for bursts, offers less maneuverability than more laterally compressed reef fish during prolonged chases. If the initial intercept fails, the predator may not easily pivot into sustained pursuit. This constraint imposes selective pressure for accuracy. The apparent dominance of speed is tempered by metabolic economics. Every strike must justify its cost. In larger individuals, solitary hunting predominates, but juveniles may form loose aggregations, learning the geometry of schooling prey. These groupings can increase confusion among sardines and anchovies, amplifying the effectiveness of individual strikes. Yet even within groups, coordination remains minimal compared to pack hunters. The barracuda's strategy is fundamentally individualistic, dependent on personal timing rather than collective choreography. The ocean's calm becomes shared staging ground for independent blades. Ecologically, barracudas occupy a mid to upper predatory tier, influencing the distribution and behavior of smaller pelagic fishes. Their presence can compress schools closer to reef structure, altering grazing patterns and nutrient flow. By selectively removing individuals, they shape genetic and behavioral traits within prey populations, favoring those with quicker reaction times or tighter schooling cohesion. The predator's silence thus drives evolutionary response, sculpting the very behaviors it exploits. Light itself becomes accomplice. In clear tropical waters, sunlight penetrates deeply, creating shafts and gradients that fragment perception. The barracuda's reflective flanks scatter this light, reducing consistent silhouette. In certain angles, the fish appears as a flash of brightness indistinguishable from surface glare. Prey navigating through these luminous corridors must pass signal from shimmer, and in that ambiguity lies the window of attack. The trap is not darkness, but dazzle. The final reversal reframes the notion of calm. What appears tranquil is charged with potential energy, and the barracuda embodies that tension. Its hovering posture is not passivity but readiness, muscles primed beneath still skin. The open water, lacking the dramatic structures of reefs or the concealment of sand, seems honest in its emptiness. Yet honesty is illusion. The predator does not need hiding places when it can align with the environment's own optical noise. As the sardine school regathers and resumes its coordinated drift, the silver line reappears at a distance, once again suspended, unreadable. The ocean hums with gentle currents and refracted light, and the space between reef and blue feels expansive and benign. Within that space, silence persists, coiled and patient. The barracuda does not vanish into sand or wedge into stone. It inhabits the interval before motion, turning the absence of obvious threat into its most reliable ally. In this borderless water, the trap is the pause itself, and the blade waits within it. The strike is already over before the water understands that it has moved. A shrimp hovers a breath above the rubble, its antennae sweeping for chemical traces of algae and detritus, its nervous system tuned to detect the forward rush of a hunting grouper or the pressure wave of an approaching snapper. It does not scan for the seabed itself. The patch of mottled coral and filamentous growth beneath it has been part of the visual field for minutes, a static irregularity against which motion would normally be detected. Then the rock opens. A cavernous mouth erupts upward in a fraction of a second, generating a localized vacuum that collapses the surrounding water column inward. The shrimp does not flee, it is hydraulically erased. By the time the surrounding plankton resettle, the reef floor has returned to stillness, and the predator has become geology once more. The animal responsible is the stonefish, a master of ecological misdirection whose body is not merely camouflaged, but structurally aligned with the architecture of the reef. Its dermal surface is a mosaic of tubercles, ridges, and fleshy appendages that mimic coralline algae and encrusting sponges, while chromatophores beneath the skin shift its tone subtly toward the prevailing palette of browns, grays, and muted greens. This is not decoration. It is an evolutionary investment in becoming statistical noise. Reef ecosystems are built on visual clutter, broken coral branches, dead skeletons, patches of cyanobacteria, and sediment drifts. So predators that announce themselves through outline or symmetry are quickly learned and avoided. the stonefish dissolves its outline into randomness, exploiting the neural tendency of prey to filter out unchanging textures. In doing so, it does not merely hide within the reef, it becomes part of the reef's informational background. Yet concealment is only half of the system. The true mechanism of its success lies in biomechanics that convert stillness into explosive force. The stonefish's cranial anatomy is designed for rapid buccal expansion. Specialized muscles depress the floor of the mouth and flare the opercula, creating a sudden drop in pressure that pulls water and prey inward with violent efficiency. This is suction feeding refined to a brutal minimum of movement. Rather than chasing, it waits for kinetic energy to drift within range, then converts stored muscular tension into an upward surge. The strike vector is often vertical, an orientation that exploits the blind zone of many reef organisms that focus laterally for approaching threats. In this geometry, gravity and surprise collaborate. The reef floor, assumed to be inert, becomes a launch platform. The narrative would seem straightforward. A patient ambush predator thriving through deception. But the first reversal is already embedded in its immobility. By surrendering speed and roaming territory, the stonefish accepts extreme metabolic constraint. It does not burn energy in pursuit, it budgets it. Its physiology is tuned for low activity and long fasting intervals, capable of surviving extended periods without a meal. In a reef where food availability fluctuates with currents and dial cycles, this metabolic austerity becomes a strategic advantage. While more active hunters must feed frequently to sustain muscle mass and territorial patrols, the stonefish can endure scarcity. What appears to be lethargy is in fact a calibrated response to ecological unpredictability. It is not lazy, it is economically ruthless. The reef, however, is not a forgiving stage. It is a three-dimensional maze of predators and competitors, and a creature that commits to stillness risks becoming substrate for others. Algae attempt to colonize its skin, small invertebrates may graze across its back. Rather than resisting entirely, the stonefish tolerates a degree of biological overgrowth, further enhancing its disguise. This is camouflage as ecosystem participation. Epibionts that settle on its surface blur the boundary between animal and environment, reinforcing the illusion that there is no organism present at all. In this way, the stonefish does not simply hide from perception, it recruits the reef's own growth processes to obscure itself. The environment completes the lie. The most infamous feature of this lie rises along its dorsal ridge, a series of stout spines connected to venom glands, capable of delivering one of the most potent envenomations in the marine realm. Unlike the upward strike of its feeding apparatus, these spines are defensive architecture. They are not launched or flicked, they stand ready. When pressure is applied, by a predator's bite or a careless foot descending from the surface, the integumentary sheath around each spine depresses, forcing venom through grooves and into the wound. The chemistry is a complex cocktail of proteins that disrupt cellular membranes, induce intense pain, and can trigger systemic shock. The stonefish does not pursue revenge. It responds to compression. In this design, the animal transforms accidental contact into punishment, turning the simple act of stepping into a potential medical emergency. Here, the second reversal emerges. The stonefish is not an aggressor seeking confrontation. It is an organism so committed to immobility that it must rely on deterrence rather than escape. Venom becomes the insurance policy for stillness. Fast swimmers evade by fleeing. Armored species resist by deflecting. The stonefish does neither. It trusts that its camouflage will prevent most encounters, and that its spines will resolve the few that slip through. The venom is thus not primarily an offensive weapon, but a structural necessity of a lifestyle built on surrendering motion. What seems like monstrous lethality is in fact the cost of radical passivity. Even its distribution across the Indo-Pacific reflects this ecological logic. Stonefish favor shallow reefs, seagrass beds, and rubble-strewn lagoons where human presence often overlaps with marine life. These are zones of high productivity, but also high disturbance. Tides shift sediments, storms rearrange coral fragments, and sunlight penetrates with fierce clarity. In such dynamic settings, the ability to masquerade as broken reef confers resilience. A storm that shatters coral creates more visual chaos, not less, expanding the field in which a stonefish can disappear. Habitat disruption that might disadvantage more specialized hunters can inadvertently amplify the stonefish's concealment strategy. The reef's violence feeds its anonymity, yet the stonefish is not invulnerable. Larger predators, including certain sharks and rays, may consume it despite the risk of spines, and humans have learned to treat envenomations through hot water immersion that denatures venom proteins. Evolution is never unilateral. Every advantage invites countermeasure. Still, within the microcosm of the reef floor, the stonefish occupies a niche defined less by dominance than by control over assumptions. Prey species are conditioned by repeated exposure to threats that move, cast shadows, or generate pressure waves. The stonefish exploits the cognitive blind spot that remains when background is trusted. Its power is epistemological. It wins because others believe they know what the seabed is. This manipulation of perception has cascading effects. By preying on small fish and crustaceans that graze algae and detritus, the stonefish participates in regulating trophic pathways that influence reef health. Remove such ambush predators, and grazer populations may swell, altering algal coverage and competitive dynamics among coral species. The stonefish's stillness thus ripples outward through trophic cascades, shaping the very substrate it imitates. It is both consumer and curator of the reef's balance. The place it weaponizes is also the place it helps maintain. The final reversal lies in our own interaction with this animal. To divers and coastal communities, the stonefish is a hazard, a lurking toxin disguised as scenery. But this framing misses the deeper truth. The stonefish does not hunt humans, it endures them. It has evolved within a sensory world where the greatest threats approach from above, casting unpredictable shadows. Its dorsal spines are oriented upward because the reef's most dangerous pressures descend. In shallow water, a human foot is simply another descending force. The envenomation that feels like targeted malice is an automated reflex in a creature that has relinquished the option of escape. We step into its geometry and trigger a defense written millions of years before our arrival. In the end, the stonefish is the purest expression of an ecological principle, that survival can be achieved not by outpacing the environment, but by dissolving into it so completely that boundaries vanish. It turns place into punishment by embedding itself in the assumptions that define safety. The reef teaches its inhabitants that rough texture is inert, that broken coral does not bite, that the seabed is foundation rather than threat. The stonefish listens to that lesson and becomes its exception. It does not roar, chase, or flare in warning. It waits, metabolically restrained, anatomically armed, and visually erased, until the neutral ground betrays those who trust it. When the water settles after another upward eruption, nothing appears altered. The rubble remains rubble. The algae drift. The reef resumes its quiet transactions of light and nutrient. Only one small absence marks the event. A prey item folded into the camouflage of its killer. In that absence lies the true mastery of the stonefish. The ability to convert invisibility into impact, to let the environment do the lying, and to transform the most ordinary patch of seabed into a threshold where assumption becomes consequence. The reef floor does not move, yet something waves from it with deliberate rhythm. A filament twitches in the current, pale and worm-like, rising from what appears to be a lump of sponge crusted with algae and sediment. A juvenile damselfish hovers nearby, its sensory world tuned to flickers of edible life, its lateral line passing minute vibrations in the water. The filament pulses again, imitating the erratic swim of injured prey. The damselfish inches closer, drawn by hunger and by a neurological bias towards small, isolated movements that signal opportunity rather than threat. The sponge erupts. A cavernous mouth yawns open with a velocity so extreme that water itself collapses inward, and the damselfish vanishes not through pursuit, but through physics. The reef returns to stillness, and the waving filament resumes its patient dance. The architect of this deception is the frogfish, a master of angling and anatomical distortion, whose body has abandoned conventional fish geometry in favor of camouflage that borders on grotesque abstraction. Its skin is thick, pliable, and studded with dermal spinules that resemble encrusting organisms, while chromatophores adjust its coloration toward the dominant hues of nearby coral rubble or sponge colonies. Unlike streamlined predators built for speed, the frogfish is compact, rotund, and supported by limb-like pectoral fins that allow it to walk across the substrate in slow, deliberate steps. This morphology is not a compromise. It is an evolutionary wager. By sacrificing hydrodynamic efficiency, the frogfish invests in remaining unreadable. It does not want to be interpreted as fish. It wants to be misclassified as reef. The deception deepens with the elysium and esker, a modified dorsal spine tipped with a fleshy lure that can be wiggled independently of the body. This apparatus is not decorative, but neurologically integrated, controlled by musculature that allows fine-scale oscillation and orientation. In ecological terms, it is a behavioral amplifier layered atop morphological camouflage. Where the stonefish relies primarily on stillness and explosive suction, the frogfish actively scripts the scene. It introduces a moving stimulus into a static field, exploiting the prey's evolved attraction to isolated, prey-like motion. Many reef fish are conditioned to approach small, erratic targets that promise caloric gain. The frogfish weaponizes that conditioning, presenting a false reward in a context that appears benign. The reef becomes not merely background but stage, and the frogfish manipulates its audience. Yet the mechanics of the kill are even more astonishing than the performance. Frogfish possess one of the fastest feeding strikes in the vertebrate world, capable of expanding their buccal cavity and engulfing prey in a fraction of a second. The jaws open and the mouth cavity balloons outward, increasing its volume dramatically and generating a negative pressure gradient that pulls prey inward along with a torrent of surrounding water. This suction is so forceful that it can capture animals nearly equal in size to the predator itself, folding them into a distensible stomach lined with elastic tissue. The frogfish does not chew. It swallows whole, relying on expandable connective structures and potent digestive enzymes to process meals that would seem anatomically impossible. In this act, the reef's clutter becomes a portal into a hydraulic trap, and scale ceases to matter. The first narrative reversal lies in this grotesque stillness. To the untrained eye, the frogfish appears deformed, almost maladapted, as though evolution abandoned elegance in favor of clumsiness. Its swollen body, lack of visible neck, and awkward locomotion suggest vulnerability rather than dominance. Yet this apparent weakness is a hyper-specialization. By minimizing movement and embracing asymmetry, the frogfish reduces the likelihood of detection by prey that scan for predictable silhouettes and rhythmic fin beats. The very features that seem to betray its identity as fish are the keys to its survival. It is not poorly designed. It is exquisitely tuned to exploit the cognitive blind spots of other reef inhabitants. This strategy unfolds within a reef ecosystem structured by complexity and competition. Coral heads, sponges, and macroalgae compete chemically and physically for space, releasing allelopathic compounds that inhibit rivals and shaping microhabitats of shadow and current. Within this mosaic, small fish navigate a labyrinth of crevices, balancing the need to forage against the risk of predation. The frogfish inserts itself into this chemical and structural chaos, aligning its body texture with nearby organisms and tolerating epibionts that further blur its outline. By embedding in zones of high structural complexity, it capitalizes on the sensory overload experienced by prey species. When every inch of substrate presents irregular shapes and potential hiding spots, the threshold for identifying danger rises. The frogfish thrives in that ambiguity. Metabolically, it is a creature of patience. Like many ambush predators, the frogfish operates under energy constraints that favor sporadic but substantial meals over constant activity. Its slow movements and extended fasting intervals reflect a physiology optimized for conserving energy in environments where prey availability fluctuates with tides, currents, and diel cycles. This austerity is not mere lethargy. It is a calculated adaptation to a habitat where bursts of abundance alternate with stretches of scarcity. When prey density spikes, the frogfish's rapid strike capitalizes on opportunity. When prey thins, its low metabolic demands allow it to endure. The reef's unpredictability becomes less a threat and more a rhythm to which the frogfish is attuned. But the illusion of total control fractures when the frogfish itself becomes visible to larger predators. Groupers, moray eels, and even octopuses may target frogfish, particularly when their camouflage falters against shifting backgrounds. The second narrative reversal emerges here. The master of disguise is not invisible to all. Its success depends on precise alignment with habitat texture and color, and sudden environmental changes, bleaching events, sedimentation, or algal blooms, can disrupt that alignment. A frogfish that once blended seamlessly into a sponge garden may stand out starkly against newly bleached coral. Camouflage is dynamic, not absolute. The same structural chaos that conceals it can, under altered conditions, betray it. In response, frogfish exhibit a capacity for color change over days to weeks, gradually adjusting pigmentation to match new surroundings. This is not instantaneous transformation, but physiological recalibration mediated by chromatophores and hormonal signals. The process underscores the frogfish's reliance on environmental feedback. It does not impose dominance on the reef. It listens to it, reshaping its appearance in response to shifting substrates. In this interplay, the frogfish is both manipulator and dependent, a predator whose identity is contingent on the living architecture around it. The most profound twist, however, lies in the lure itself. What appears to be a simple imitation of prey is in fact a product of co-evolutionary arms races. In some species, the esker resembles specific organisms common to their habitat, such as small worms or shrimp. Prey species evolve discrimination to distinguish genuine food from deceptive signals, while frogfish refine their mimicry in response. This feedback loop drives increasing specificity, turning the reef into a theater of sensory deception where signals are contested and meaning is unstable. The frogfish's lure is not merely bait. It is a dynamic node in a network of evolutionary pressures, shaping perception and response across multiple species. In swallowing prey nearly its own size, the frogfish momentarily abandons the illusion of inert substrate and reveals its monstrous capacity. Its jaws distend, its body flexes, and the reef witnesses a transformation from sponge-like lump to gaping maw. Yet once the prey is secured, the predator contracts, returning to its false identity. The violence is transient, the disguise is enduring. This oscillation between eruption and erasure defines the frogfish's ecological role. It punctuates the reef's steady transactions with moments of abrupt removal, thinning populations of small fish and influencing the flow of energy upward through the trophic web. By regulating certain prey species, frogfish indirectly shape grazing pressure on algae and invertebrates, contributing to the delicate balance that sustains coral growth. Remove such ambush predators, and prey populations may surge, altering competitive dynamics and potentially destabilizing sections of the reef. The frogfish's stillness thus reverberates beyond individual kills. It is an unseen hand adjusting the tempo of consumption and growth, a silent arbiter embedded within the substrate it imitates. In the final analysis, the frogfish does not merely hide in the reef. It redefines the concept of habitat. It transforms passive terrain into active machinery, converting visual clutter into a loaded mechanism primed by neurological bias and hydraulic force. The prey believes it is navigating a mosaic of coral and sponge, guided by instincts honed over generations. Instead, it drifts toward a device designed to exploit those very instincts. The frogfish is not a predator lurking behind scenery. It is scenery rewritten as predator. When the water calms after another suction strike, the reef appears unchanged. The lure resumes its subtle motion, the body settles into its counterfeit stillness, and the surrounding fish continue their foraging routes, unaware that the map they trust has been altered.
In that quiet persistence lies the frogfish's triumph, the ability to rig the game without announcing that it has begun, to embed lethal intent within the ordinary textures of the reef, and to remind us that in the ocean's most intricate habitats, the difference between background and predator can vanish in less than a heartbeat. The tunnel collapses inward without warning. A cardinal fish darts toward a narrow coral fissure to escape the advancing shadow of a trevally, its lateral line registering the pressure wave of pursuit, its survival algorithm directing it toward darkness as refuge. The crack appears stable, a geometry of limestone carved by parrotfish bites and centuries of bioerosion. Then the darkness surges forward. A head armed with recurved teeth explodes from the cavity, jaws clamping down with lateral force that arrests the fish mid-flight. In a second, the fissure ceases to be architecture and becomes anatomy. The cardinal fish is dragged backward, swallowed not by open water, but by a hole that was never empty. The reef resets into quiet complexity, yet the maze has just demonstrated that some corridors are mouths. The moray eel is engineered for this conversion of shelter into weapon. Its elongated muscular body is laterally compressed just enough to slide into crevices that are structurally inaccessible to bulkier predators, while its reduced pectoral fins eliminate protrusions that might betray its presence or limit entry into tight spaces. Unlike the streamlined swimmers of the open water column, the moray is a creature of constraint, thriving where the reef's architecture narrows and shadows deepen. Its skin, thick and coated in a mucus layer that reduces abrasion and resists microbial colonization, allows it to press against rough coral without injury. In ecological terms, it does not merely inhabit the reef, it colonizes the negative spaces within it, transforming cavities into strategic strongholds. The jaws are the first visible instruments of this strategy, but the true genius lies deeper. Morays possess a second set of jaws, pharyngeal jaws, situated in the throat, capable of extending forward to grasp prey and draw it inward. This mechanism compensates for the eel's inability to generate the powerful suction used by many bony fishes. Because their skull structure limits rapid buccal expansion, morays rely on biting rather than vacuum capture. The pharyngeal jaws, armed with additional teeth, act as a conveyor, securing prey and pulling it deeper into the esophagus. Once the initial bite is delivered at the threshold of the crevice, escape is no longer a matter of distance, but of anatomy. The prey is seized twice, first by the outer jaws and then by the hidden inner apparatus that ensures finality within the confines of stone. The reef is a labyrinth of competing survival strategies. Small fishes memorize escape routes among branching corals and rubble fields, constructing mental maps that associate certain holes with safety. Predators exploit these patterns, and the moray's approach is uniquely parasitic on the very concept of refuge. By occupying crevices that function as default escape paths, it inverts the logic of shelter. What appears to be an exit becomes a trap. This is not random opportunism, but spatial predation and exploitation of the reef's topology. In a habitat defined by structural complexity, control over choke points can yield disproportionate advantage. The moray does not patrol widely because it does not need to. The reef funnels prey toward it. Its behavior reflects this reliance on ambush and positional dominance. Morays often protrude only their heads from holes, mouths agape in a manner that might seem aggressive. In truth, this gaping is primarily respiratory, facilitating water flow over the gills in the absence of continuous swimming. The open mouth, however, doubles as intimidation, revealing rows of teeth that discourage would-be predators or competitors from probing too deeply into the crevice. Here, the first narrative reversal unfolds. The exposed head appears reckless, yet it is an adaptive compromise between respiration and defense. What looks like threat display is physiological necessity, repurposed as warning. The moray's sensory world further sharpens its command of confined spaces. Vision in these dim recesses is secondary to chemoreception. Highly developed olfactory organs detect dissolved chemical cues, allowing the eel to sense injured or stressed prey even when visibility is limited. A wounded fish releasing alarm substances into the water column creates a chemical trail that seeps into cracks and tunnels. The moray reads this invisible script and positions itself accordingly, aligning its body with currents that deliver information.
In this way, the reef's water flow becomes a communication network, and the moray interprets it with precision, born of specialization. Yet the moray is not invincible within its chosen domain. Larger predators such as reef sharks and groupers may attempt to extract it from crevices, and octopuses, themselves masters of narrow spaces, can engage in lethal contests with eels. The second narrative reversal emerges in these encounters. The eel's elongated form, optimized for entering tight spaces, can become liability if the cavity is breached or if the eel is forced into open water. Away from the structural support of the reef, it is less maneuverable and more exposed. Its dominance is conditional, contingent upon maintaining control of the whole. The predator of corridors becomes vulnerable when corridors vanish. In some regions, morays engage in cooperative hunting with groupers, a phenomenon that complicates the image of solitary ambusher. Groupers patrol the open reef, flushing prey toward crevices, while morays probe into holes that groupers cannot access. The two species signal one another through body language, effectively combining complementary hunting strategies. This alliance reveals another twist. The moray, often imagined as isolated and territorial, participates in interspecific collaboration that enhances hunting success for both partners. The reef's maze becomes a coordinated battleground where one predator drives and the other intercepts. The solitary mouth in the wall is part of a broader network of predatory intent. Metabolically, the moray reflects the trade-offs of ambush life. It does not expend constant energy in pursuit, allowing it to endure periods between successful captures. Its musculature, however, must deliver powerful bursts to seize prey at the threshold of a crevice, and its digestive system must process meals that may be relatively large compared to its body diameter. The eel's flexible skeleton and absence of rigid rib cages facilitate this capacity, enabling it to ingest prey that seems disproportionate to the width of its head. Once swallowed, the prey is digested slowly, energy extracted over time, sustaining the eel during intervals of stillness. The reef's unpredictability is buffered by physiological patience. The broader ecological implications of the moray's strategy ripple outward. By preying on small fishes and crustaceans that graze algae or consume invertebrates, morays influence the balance of reef communities. Their presence can modulate prey populations, indirectly affecting algal growth and coral competition. In ecosystems where predator diversity declines, the absence of morays may shift pressure onto certain prey species, altering trophic cascades that reverberate through the coral architecture. The hole that becomes a mouth is not merely a localized hazard, it is a node in the reef's regulatory web. There is also a chemical dimension to the moray's existence. The mucus coating its skin contains compounds that reduce parasite loads and prevent abrasion in tight quarters. This biochemical shield allows it to inhabit spaces where constant friction would injure other fishes. What might appear as slimy residue is in fact adaptive secretion, a protective interface between body and stone. The reef's abrasiveness, lethal to the unprotected, becomes tolerable terrain for the eel. Weakness is converted into advantage through chemistry. In the end, the moray eel does not dominate the reef through speed or spectacle. It weaponizes absence, uncertainty, and geometry. It turns cavities into claims and shadows into territory. For every fish that memorizes a safe passage, there exists the possibility that the passage is occupied by muscle and teeth. The ocean becomes a maze in which architecture is not neutral. Some openings inhale. When the cardinal fish vanishes into the fissure and the water resumes its gentle oscillation, the reef appears unchanged, its corals branching, its sponges filtering, its currents weaving between structures. Yet the map has been redrawn by presence concealed within voids. The moray remains, head protruding, jaws parting and closing in slow respiration, a reminder that in complex systems, emptiness can be illusion, and refuge can conceal a predator patient enough to let the maze deliver its next victim. A larval fish drifts too close and the water itself detonates. Microscopic harpoons fire in a synchronized discharge so fast that nerve impulses cannot outpace them, and in the span of a blink, the translucent body is pinned by threads laced with venom. The tentacles that moments ago seemed ornamental now contract with muscular precision,
guiding the immobilized prey toward a central mouth that opens not in pursuit, but in response. The sea anemone has not moved from its anchor point on rock, yet the boundary around it has proven lethal. The beauty was never benign. It was perimeter defense, written in color and motion. The sea anemone is a cnidarian, an animal whose tissues are organized around radial symmetry and a single opening that functions as both mouth and exit. Yet its simplicity belies a biochemical arsenal of staggering sophistication. Each tentacle is studded with nematocysts, specialized stinging organelles housed within cnidocytes that respond to mechanical and chemical cues. When triggered, these microscopic capsules explosively evert, driving barbed threads into the target and injecting toxins that disrupt cellular membranes and interfere with ion channels. The process is mechanical and chemical at once, a ballistic event powered by osmotic pressure that rivals the acceleration of far larger biological systems. The anemone does not pursue. It establishes a hazard gradient in the water column, transforming proximity into risk. Anchored by a pedal disc that adheres to substrate through adhesive secretions and muscular contraction, the anemone occupies high traffic zones where currents deliver plankton and suspended organic matter. Reef hydrodynamics become its conveyor belt. In regions where tidal flows pulse predictably, the anemone extends its tentacles into the stream, increasing surface area for interception. This is not random waving but strategic positioning within the reef's fluid architecture. The currents that nourish coral polyps and carry oxygen to benthic communities also ferry unsuspecting organisms into the anemone's reach. In this way, the animal converts environmental motion into feeding opportunity without ever abandoning its post. Yet the most striking aspect of the anemone's existence is the paradox of visibility. Unlike ambush predators that depend on concealment, the anemone is conspicuous. Its tentacles shimmer with pigments that can range from muted earth tones to brilliant greens and reds, often enhanced by symbiotic algae known as zooxanthellae residing within its tissues. These photosynthetic partners contribute energy through carbon fixation, supplementing the anemone's diet and linking it directly to the reef's primary productivity. The tentacles that sting are also solar collectors, their translucence optimized for light penetration. What appears decorative is metabolically purposeful, integrating predation and photosynthesis into a single waving structure. The first narrative reversal emerges in this symbiosis. The anemone, rooted and seemingly passive, is not solely a carnivore awaiting drifting prey. It is also a host in a mutualistic partnership that blurs the boundary between animal and plant-like function. During daylight hours, its internal algae convert sunlight into sugars, offsetting the energetic cost of maintaining stinging cells and muscular responses. This dual strategy allows the anemone to persist in nutrient-poor waters where prey capture alone might not suffice. Apparent fragility becomes resilience through collaboration. The hazard zone is powered partly by the sun, but the tentacle's sting is not indiscriminate. Certain fish species, most famously clownfish, navigate these lethal filaments unharmed, weaving among them with agility that would doom other organisms. The secret lies in a mucus coating that prevents the discharge of nematocysts, either by mimicking the anemone's own chemical signature or by inhibiting the mechanical trigger threshold. Through careful acclimation, the fish integrate into the anemone's perimeter, gaining protection from predators that dare not penetrate the stinging field. Here the second narrative reversal unfolds. The anemone's border, lethal to most, becomes sanctuary to a select few. What is trapped to one species is refuge to another, and the hazard zone transforms into a shared fortress. This mutualism alters the reef's social dynamics. Clownfish defend their host anemone aggressively, chasing away butterfly fish and other predators that might nibble on tentacles. Their movements enhance water circulation around the anemone, increasing oxygenation and potentially improving waste removal. In return, the anemone provides shelter and, at times, food scraps from captured prey. The rooted organism becomes the nucleus of a micro-community, its stinging perimeter structuring interactions among species. The waving tentacles are not only weapons, but walls around a living citadel. Beyond symbiosis, the anemone participates in the chemical contests that define reef ecosystems. Corals and anemones compete for space on hard substrate, deploying sweeper tentacles or allelopathic compounds to inhibit rivals. When territories overlap, contact can result in localized tissue damage and retraction, a slow-motion battle for centimeters of real estate. The anemone's nematocysts are thus tools of both predation and territorial defense, shaping spatial patterns on the reef. The rock to which it clings is not inert ground, but contested territory in a landscape where sessile organisms wage silent wars. Metabolically, the anemone operates under constraints imposed by its stationary lifestyle. It cannot relocate rapidly in response to environmental change, though some species can detach and crawl short distances using muscular contractions of the pedal disc. Its energy budget must accommodate the maintenance of nematocysts, the digestion of prey within a gastrovascular cavity, and the metabolic demands of symbiotic algae. During periods of stress, such as elevated water temperature or pollution, the relationship with zooxanthellae can break down, leading to bleaching events that strip the anemone of its photosynthetic partners. In such moments, the brilliant hazard fades, revealing vulnerability beneath the display. The final reversal lies in the perception of softness. To human eyes, the anemone resembles a flower swaying in a garden, delicate and inviting. Yet its tentacles represent one of the ocean's most effective contact-based defense systems, capable of deterring fish many times its size. The lack of bones or rigid structures does not equate to weakness. Instead, hydrostatic support and muscular fibers allow the anemone to retract swiftly into a compact form when threatened, minimizing exposure. The very softness that suggests fragility enables flexibility and rapid response. What seems ornamental is engineered for survival. As currents shift and plankton pulses through the reef, the anemone continues its patient vigil. Each passing organism must interpret the waving tentacles and decide how close is too close. Some misjudge and are captured in a flash of microscopic violence. Others, adapted through coevolution, slip safely among the filaments and find shelter where predators hesitate to follow. The trap is visible, even radiant, yet still misread because its beauty masks its function. In this ecosystem of intricate negotiations, the sea anemone stands as both sentinel and snare. It does not chase the world. It conditions the space around it so that the world's own movements determine fate. The reef is a tapestry of motion and color, but within that tapestry certain threads are charged with venom and sunlight alike. The anemone's tentacles continue to wave, inviting interpretation, reminding every drifting creature that in the ocean's architecture, decoration can be defense, and stillness can conceal a perimeter where contact becomes consequence. The predator commits to the chase and seals its own mistake. A young wrasse bolts across the reef flat, scales flashing, pursued by a snapper whose jaws are built for decisive closure. The wrasse aims not for open water, but for a cluster of waving tentacles that shimmer like a floral ornament in the current. The snapper calculates speed and angle, unaware that the target is a corridor lined with microscopic weapons. The wrasse vanishes into the tentacular forest. The snapper lunges through the same opening and recoils as nematocysts detonate along its snout and gill covers, venom saturating contact points in a cascade of cellular disruption. The fortress was visible all along. Only one species holds the key. The clownfish's advantage is biochemical rather than muscular. Its skin secretes a mucus layer that either lacks the chemical triggers that provoke nematocyst discharge or actively suppresses them, allowing the fish to weave through tentacles that would immobilize nearly any other small reef inhabitant. This immunity is not instantaneous. Juvenile clownfish perform a careful acclimation ritual, brushing tentacles in controlled contact to incorporate the anemone's chemical signature into their mucus. Through this process, the fish effectively rewrites the anemone's recognition system, becoming indistinguishable from host tissue at the molecular interface. The passcode is not strength, it is compatibility. The anemone itself is a stationary arsenal, armed with stinging cells powered by osmotic pressure that fire with mechanical precision upon stimulation. To most reef fish, it is a no-go zone, a chemically defended perimeter that enforces spatial segregation. The clownfish converts that exclusion into ownership. By inhabiting the tentacles, it claims territory that others cannot contest.
Predators that rely on rapid pursuit are forced to calculate the cost of entry, and most withdraw after initial contact with the stinging barrier. The clownfish's small size and bright coloration, which would ordinarily advertise vulnerability, become paradoxical signals within this context. The reef reads the colors as prey, yet the territory reads them as authorized presence. This is fortress living in its purest form. The clownfish does not need to outswim larger fish or outcompete them in open water. It leverages the anemone's chemical minefield as defensive infrastructure, turning a hazardous border into private real estate. The relationship is mutualistic rather than parasitic. The fish defends its host from anemone-eating butterflyfish and other intruders, increasing the anemone's survival odds. Its movements enhance water circulation among tentacles, improving gas exchange and nutrient distribution. Waste products excreted by the fish supply nitrogen compounds that fertilize the symbiotic algae within the anemone's tissues, boosting photosynthetic output. The fortress is not merely occupied, it is maintained and provisioned. The first narrative reversal lies in perception. The clownfish appears fragile, a small-bodied creature with limited speed and modest dentition. In open water, it would rank low in the hierarchy of reef combatants. Yet by choosing to live within a stinging labyrinth, it elevates its strategic position. Weakness becomes leverage. The bright colors that might attract predators function instead as honest signals of toxicity-adjacent territory. Predators learn through painful experience that pursuing such prey carries disproportionate risk. The clownfish's apparent vulnerability becomes a calculated display backed by environmental enforcement. Within each anemone, social structure adds another layer of complexity. Clownfish live in hierarchical groups dominated by a breeding female, the largest individual, followed by a breeding male and several non-breeding subordinates. Should the female perish, the breeding male undergoes sex change, transforming into a functional female, while the largest subordinate matures into the new breeding male. This protandrous hermaphroditism ensures reproductive continuity within the fortress, eliminating the need for risky dispersal in search of mates. The anemone becomes not only shelter, but generational anchor, a fixed point around which life cycles revolve. This reproductive strategy is tightly coupled to territorial defense. The dominant female aggressively repels intruders, including other clownfish species, maintaining exclusive access to the host. The limited number of suitable anemones in a reef system imposes strong selection pressure on competition and cooperation. The fortress is finite real estate, and occupancy confers survival advantage. Here, the second narrative reversal emerges. What appears to be a tranquil partnership is in fact a site of intense social negotiation, where size, rank, and timing determine reproductive fate. The protective perimeter that shields the group from external threats also concentrates internal dynamics. Ecologically, the clownfish-anemone partnership influences local community structure. Predators that avoid anemones may shift hunting routes, altering patterns of prey movement. Grazing fish navigating reef topography must account for stinging zones, creating microhabitats of relative safety for certain species and exclusion for others. The fortress thus radiates effects outward, shaping traffic flow and trophic interactions in subtle ways. In a system governed by spatial complexity, the placement of a single anemone can alter behavioral maps across square meters of reef. Metabolically, the clownfish benefits from the stability of its chosen habitat. By reducing the need for constant roaming, it conserves energy that would otherwise be spent on evasion and exploration. Its diet consists of small invertebrates, plankton, and food scraps captured by the anemone. The proximity to a constant shelter reduces mortality risk, allowing energy allocation toward growth and reproduction rather than perpetual flight. The fortress reduces uncertainty, and in evolutionary terms, reduced uncertainty translates into fitness. Yet the arrangement is not immune to environmental stress. Rising sea temperatures and ocean acidification threaten both coral reefs and anemone hosts. Bleaching events that expel symbiotic algae from anemones diminish their health and structural integrity. Without a robust host, the clownfish loses its sanctuary. The fortress crumbles not through invasion, but through climate-driven destabilization. In this broader context, the clownfish's leverage appears fragile, contingent upon the persistence of its chemical ally. The final reversal confronts the notion of ownership. The clownfish seems to control its territory, yet its existence is inseparable from the anemone's biology. The passcode is valid only so long as the host maintains functional nematocysts and symbiotic balance. The fish's dominance is borrowed from a stationary organism rooted to rock. In claiming the fortress, the clownfish is simultaneously dependent upon it. Power and vulnerability intertwine. As the snapper retreats from the stinging perimeter, its sensory systems recalibrating after the shock, the clownfish resumes its patrol among the tentacles, unscathed. The reef continues its complex choreography of currents, grazers, and predators. Yet within one patch of waving filaments, a small fish has rewritten the rules of engagement. It has transformed hazard into home, barrier into advantage, and bright vulnerability into territorial command. The trap belongs to the environment, but the clownfish has learned to live at its center, turning a biological minefield into a private citadel where size is irrelevant and location is everything. The seal feels the impact before it sees the shape. A torpedo of muscle and cartilage erupts from the black beneath, jaws yawning wide as white water detonates against the surface. For a fraction of a second, gravity is suspended. The seal is airborne, the ocean torn open by force traveling upward through a column of water that only moments ago looked featureless and calm. Then both bodies crash back into the sea, and the vertical corridor closes as if nothing passed through it at all. The empty blue was never empty. It was depth waiting for timing. The great white shark is not merely a predator of open water, it is an engineer of the light gradient. In the pelagic realm, where reef architecture is absent, structure is defined by physics rather than stone. Sunlight penetrates from above, attenuating with depth, creating a sharp contrast between the glare of the surface and the obscurity below. Seals and sea lions swimming near the top occupy a zone of relative brightness, their eyes tuned to scan horizontally and upward for threats. The space directly beneath them, however, is a blind wedge, where light fails and silhouettes blur. The great white aligns itself within this optical boundary. Its counter-shaded body, dark dorsally, pale ventrally, rendering it indistinct against both abyss and glare. Camouflage here is not about coral mimicry, but about becoming the gradient itself. Counter-shading functions as optical deception in 3 dimensions. When viewed from above, the shark's darker back merges with deeper water. When seen from below, its lighter underside blends with the illuminated surface. This adaptation, shared by many pelagic predators, reaches extreme utility in the great white because of its size and preferred hunting tactic. By approaching from depth and accelerating vertically, the shark compresses the prey's decision window to a heartbeat. Reaction time depends on detection, and detection depends on contrast. In the half-lit corridor between brightness and shadow, contrast dissolves. The seal must trust that the void is safe because its sensory systems cannot resolve what hides there. The great white's body is tuned for explosive ascent. Though lacking a swim bladder, it regulates buoyancy through a massive liver rich in low-density lipids, reducing the energetic cost of maintaining position in the water column. Its caudal fin, crescent-shaped and powerful, delivers thrust optimized for sudden acceleration. Beneath its skin lies a network of red muscle fibers, capable of sustained cruising, interlaced with white muscle fibers that generate rapid bursts. This dual musculature allows the shark to patrol at moderate speed, conserving energy, and then unleash extraordinary velocity when prey drifts overhead. The ocean's depth is not passive space. It is stored kinetic potential. Electroreception adds another layer of precision. Ampullae of Lorenzini, gel-filled pores concentrated around the snout, detect minute electrical fields generated by muscle contractions in prey. Even if visual cues are limited by turbidity or glare, the shark can sense the bioelectric signatures of a seal paddling at the surface. This sensory apparatus transforms the water column into a map of invisible currents and impulses. The prey's own physiology betrays it, broadcasting location through faint electrical whispers that the shark interprets with neural acuity. The first narrative reversal lies in the apparent emptiness of open water. Compared to a reef's labyrinthine complexity, the pelagic zone seems exposed and structureless. Yet the absence of coral does not mean absence of geometry. Depth itself is architecture, and light attenuation is wall and ceiling. The shark exploits this invisible construction, converting a vertical gradient into an ambush corridor more lethal than any crevice. The emptiness is a facade. The true structure is photic physics. When the shark strikes, the surface becomes constraint. Seals resting at the air-water boundary have limited escape vectors. They cannot dive downward quickly enough to outrun a predator ascending at speed, and lateral movement is curtailed by the sudden eruption beneath them. The surface, which provides access to air and vantage point, becomes trapdoor. In many attacks, the shark breaches entirely, its mass propelled above the waterline, jaws closing mid-air. This behavior is not theatrical flourish, but biomechanical necessity. To generate sufficient force to incapacitate agile prey, the shark must commit to a trajectory that sometimes carries it into the atmosphere. The ocean's boundary is not barrier, but launch pad. Yet the great white does not indiscriminately consume everything it bites. Research into predatory patterns suggests that some attacks on humans are exploratory, a single bite followed by release, possibly because the energetic payoff of such prey does not match that of fat-rich marine mammals. This introduces the second reversal. The image of mindless aggression gives way to calculated energy economics. The shark's feeding strategy is governed by caloric return relative to risk and expenditure. Marine mammals with dense blubber layers provide high-energy rewards that justify the burst of acceleration and exposure inherent in breaching. In ecological terms, the shark is optimizing fuel efficiency in a vast environment where energy acquisition dictates survival. The relationship between predator and prey shapes coastal ecosystems. In regions where great whites frequent seal colonies, prey species alter behavior, avoiding certain haul-out sites or adjusting foraging times. This predator-induced landscape of fear can cascade through trophic levels, influencing fish distribution and even kelp forest dynamics. By regulating pinniped populations and modifying their spatial patterns, great whites participate in maintaining ecological balance. Remove the apex predator, and mesopredator populations may surge, destabilizing food webs that evolved under its pressure. The shark's vertical ambush reverberates horizontally across the ecosystem. The great white's physiology reflects evolutionary investment in longevity and resilience. With slow growth rates and late maturity, it occupies a life history strategy that prioritizes survival over rapid reproduction. This makes populations vulnerable to overfishing and habitat disruption. The predator that commands the invisible half of the ocean is itself susceptible to human-induced change. Overexploitation can thin numbers to levels where ecological roles diminish, altering the balance between predator and prey in coastal systems. Thermoregulation adds another dimension to its mastery. Unlike most fish, the great white exhibits regional endothermy, maintaining elevated temperatures in certain muscle groups through countercurrent heat exchange. This adaptation allows for enhanced power output and neural processing speed in cooler waters, expanding hunting range into temperate zones where seals congregate. Warmed muscles contract more efficiently, sharpening the precision of upward strikes. The shark's internal furnace transforms cold gradients into opportunity. In the end, the great white shark does not simply own depth. It integrates physics, physiology, and perception into a seamless, predatory calculus. The seal that scans the horizon for dorsal fins must also reckon with what lies beneath, though it cannot truly see it. The ocean's vertical dimension is a theater of uncertainty, where trust in emptiness can be fatal. The shark occupies that uncertainty, shaping it, reading it, and striking through it with force that seems supernatural, but is grounded in anatomy and thermodynamics. As the disturbed surface settles and foam dissipates, the sea regains its placid facade. Light refracts in gentle ripples, and the horizon appears unbroken. Yet beneath that shimmer, Depth remains a corridor owned by muscle and instinct, a realm where the gradient between brightness and shadow conceals the calculus of a predator that has mastered the art of turning empty space into lethal structure. The water is already clouded when the mullet school fractures. Silt hangs in suspension over a shallow seagrass flat, stirred by wind and tidal shift, turning sunlight into diffuse glow rather than sharp beam. The fish cannot see more than a few body lengths ahead, and their lateral lines vibrate with the confused echoes of one another. Then the haze condenses into mass. A broad head emerges from the blur, striped flanks ghosting between green and brown, jaws opening with deliberate force rather than explosive theatrics. The school does not explode in one direction because there is no clear direction to choose. The tiger shark does not need invisibility. It needs indecision. Unlike predators that specialize in singular controlled strike environments, the tiger shark operates in coastal borderlands where clarity is rare and boundaries are unstable. Estuaries, reef edges, mangrove channels, drop-offs, and sand flats intersect in mosaics that change with tide, season, and storm. In these waters, turbidity reduces visual acuity for prey species accustomed to scanning horizons. The tiger shark thrives in this environmental ambiguity, its sensory systems built not for perfect staging, but for constant recalibration. Where other sharks favor open pelagic corridors or structured reef slopes, the tiger shark patrols transition zones, exploiting the cognitive lag that murky water imposes on smaller animals. Its anatomy reflects versatility. The tiger shark's broad, blunt head houses large olfactory organs capable of detecting faint chemical trails in diluted water, while its ampullae of Lorenzini register bioelectric pulses even when visibility collapses to near nothing. In sediment-rich estuaries, where visual contrast dissolves into monotone haze, scent and electricity become dominant channels of information. The shark reads turbulence and chemistry as others might read light and silhouette. This sensory plasticity converts uncertainty into navigational map, allowing it to move confidently where prey hesitate. The dentition is equally adaptable. Tiger sharks possess large, serrated teeth with pronounced notches, capable of slicing through turtle carapace, fish bone, and mammalian flesh. This morphological generalism supports a diet that is famously eclectic, ranging from sea turtles and seabirds to crustaceans and carrion. In ecosystems defined by shifting prey availability, dietary flexibility is insurance against scarcity. The tiger shark does not wait for a single prey type to align perfectly with its strike geometry. It samples broadly, adjusting predatory focus to what the environment offers. In doing so, it stabilizes its own energy intake within habitats that fluctuate in productivity. The first narrative reversal lies in this opportunism. To some, the tiger shark's wide-ranging diet and patrol behavior might appear less refined than the pinpoint ambush of a great white or the cooperative precision of pack hunters. Yet this very breadth is specialization for unpredictability. Coastal borderlands are dynamic interfaces where fresh water mixes with salt, sediment loads spike after rain, and prey assemblages shift with spawning cycles. A predator that commits to narrow tactics risks starvation when conditions swing. The tiger shark's apparent lack of selectivity is in fact strategic resilience. It is not indiscriminate, it is adaptive. Within seagrass meadows, the shark's presence shapes prey behavior profoundly. Sea turtles grazing on vegetation alter feeding patterns when tiger sharks frequent the area, reducing time spent in exposed shallows and shifting to safer, though less optimal, patches. This predator-induced habitat modification cascades through the ecosystem. Reduced grazing pressure in certain zones can allow seagrass to flourish, stabilizing sediment and supporting invertebrate communities. The tiger shark's patrol thus influences plant biomass and nutrient cycling, illustrating how a single predator can modulate entire coastal systems through fear alone. Yet the murk that aids the tiger shark also conceals threats. In turbid estuaries, human activity intersects with shark territory, and fishing gear can ensnare even these formidable animals. Overfishing reduces prey abundance, forcing sharks to expand patrol ranges and increasing energetic expenditure. The second narrative reversal emerges here. The master of ambiguity is vulnerable to anthropogenic clarity.
nets and hooks, invisible to human eyes until hauled, operate within the same murky borderlands, transforming the decision trap back upon its architect. The tiger shark's reliance on transitional habitats makes it susceptible to human alteration of those very zones. Metabolically, the tiger shark balances cruising endurance with bursts of power. Its large pectoral fins provide lift, minimizing energy spent counteracting negative buoyancy, while its muscular tail generates steady propulsion across long coastal stretches. Unlike breaching specialists, the tiger shark often approaches prey laterally or from below with less dramatic vertical eruption, conserving energy for sustained patrol. In uncertain water, prolonged presence can be more effective than singular spectacle. The shark becomes a moving boundary condition, its silhouette shifting between sand and shadow. The visual patterning that gives the tiger shark its name, dark vertical bars along flanks, serves as disruptive camouflage in dappled coastal light. Against backgrounds of mangrove roots and patchy seagrass, these stripes break up body outline, complicating recognition at intermediate distances. In shallows where sunlight flickers through wavelets and vegetation, the shark's pattern merges with visual noise. Camouflage here is not about matching a uniform background, but about fragmenting shape within complexity. The coastal borderland is a realm of edges. Fresh water meets salt water, sand meets reef, light meets turbidity. Edges create ecological richness, but also perceptual challenge. Prey species navigating these gradients must constantly update threat assessments. The tiger shark capitalizes on moments when assessments lag behind environmental change. A fish accustomed to clear reef water may misjudge risk upon entering silty estuary. A turtle grazing confidently in calm shallows may not detect a predator approaching through glare and sediment plume. The shark thrives in the seconds where certainty dissolves. The final reversal reframes the notion of chaos. The murky borderland appears disorderly, lacking the aesthetic clarity of coral gardens or open blue expanses. Yet, within this seeming disorder lies pattern that the tiger shark understands intimately. Tidal rhythms, spawning aggregations, and seasonal migrations create predictable pulses within the mess. The shark's wide-ranging patrols intersect these pulses, aligning with times and places where prey density spikes. What seems random to the casual observer is structured opportunity to a predator attuned to ecological tempo. As the mullet school regroups and sediment begins to settle, the tiger shark continues its glide along the shifting margin, unhurried and deliberate. The water remains imperfect, the visibility compromised, the boundaries blurred. In this borderland, hesitation is common and misreading frequent. The shark does not need a single concealed pitfall or a precisely staged ambush corridor. It inhabits a world where the entire landscape is provisional, where every patch of haze carries possibility. The trap is not a spot on the seabed. It is the uncertainty itself, and the tiger shark moves through it with the quiet authority of an animal that has made ambiguity its advantage. The rock shifts, but only after the crab is already committed. A narrow crevice at the reef base looks like routine shelter, a dark slit between limestone plates where a spiny leg can wedge safely while the tide turns. The crab advances sideways, antennae testing current, unaware that the interior wall of the crack is breathing. The surface puckers, changes tone from mottled stone to granular sand, then liquefies. 8 arms uncoil without warning, suckers sealing against carapace in a silent vacuum of flesh and calcium. The crevice was not empty. It was edited. The octopus does not merely occupy shelter, it customizes it. Its body is almost entirely muscle and connective tissue wrapped around a beak and a ring of neurons that rival some vertebrates in processing complexity. Lacking bones and external armor, the octopus can compress its mantle and funnel through apertures scarcely wider than its eye, exploiting the only rigid constraint it cannot deform. This anatomical plasticity allows entry into cracks that appear structurally insignificant to other reef inhabitants. The geometry of rock becomes negotiable, not fixed. What is refuge to a crab is workspace to an octopus. Chromatophores, iridophores, and leucophores embedded in its skin
respond to neural signals with astonishing speed, altering color, reflectance, and texture. Papillae rise and fall, transforming smooth flesh into a facsimile of coral rubble or algae crust within seconds. This is not simple camouflage. It is dynamic habitat integration. The octopus reads substrate pattern and light angle through sophisticated visual processing, then reconfigures its exterior to match context. The environment becomes canvas, and the animal becomes brushstroke. In this fluid exchange, detection thresholds blur. Predators scanning for outline and symmetry encounter a living anomaly that refuses to hold a stable shape. Yet the true misdirection lies not in appearance alone, but in manipulation. Octopuses are known to collect shells, stones, and discarded fragments, arranging them at den entrances to create barricades or partial walls. This behavior reflects foresight and spatial reasoning, traits supported by a distributed nervous system in which a significant portion of neurons reside within the arms themselves. Each limb can execute complex movements semi-autonomously, exploring crevices, testing textures, and coordinating grip. The den becomes architecture curated by intelligence. A hole in rock evolves into a controlled chamber with limited entry points and defensive leverage. The first narrative reversal challenges the notion of softness as weakness. Without bones or spines, the octopus appears vulnerable, an animal exposed to teeth and claws. Yet its softness is strategic flexibility. By conforming to the environment, it eliminates rigid leverage points predators might exploit. A moray eel biting into an octopus does not encounter solid resistance, but elastic retreat, flesh slipping deeper into cracks that eel bodies cannot follow. The apparent fragility is adaptive evasion. Softness becomes shield. Hunting strategy extends this environmental editing. When targeting crabs or bivalves, the octopus often blocks alternate exits with arms, sealing gaps before delivering a decisive bite with its beak. Some species drill small holes into shells using radula scraping and salivary secretions that weaken calcium carbonate, injecting venom to relax adductor muscles. The process is meticulous, combining chemical and mechanical tactics. The prey's own shelter becomes liability, a confined space optimized by the octopus for extraction. The trap is not ambush in open water, but orchestration within microarchitecture. Cognition amplifies these tactics. Laboratory and field observations demonstrate problem-solving abilities, memory retention, and even play behavior. An octopus encountering a novel object can investigate, manipulate, and incorporate it into den structure or discard it as irrelevant. This capacity to experiment enhances survival in dynamic reef environments where storms rearrange substrate and human debris introduces unfamiliar materials. Plastic bottles, coconut shells, and other artifacts may become temporary shelters or barriers. The animal's intelligence extends habitat rather than merely responding to it. The second narrative reversal emerges when the octopus itself becomes target. Sharks, groupers, and dolphins prey upon octopuses, exploiting moments when camouflage fails or when the animal ventures too far from den. In such encounters, the octopus deploys ink, a cloud of melanin-rich secretion that creates visual smokescreen and chemical confusion. The ink may form pseudomorphs, denser blobs that resemble the octopus's shape, diverting predator attention. The environment again becomes stage, but now the octopus edits perception by adding false cues. The predator lunges at phantom, while the real body contracts into fissure. Intelligence becomes escape art. Metabolically, the octopus is bound by rapid growth and short lifespan. Many species mature quickly and reproduce once before senescence, a life history strategy that prioritizes swift accumulation of biomass and neural development. The energetic cost of maintaining large neural tissue and dynamic camouflage is high, requiring efficient predation. Failure to secure food within edited habitat means swift decline. Intelligence is expensive, and the octopus pays for it daily through calculated hunting. In broader ecological context, octopuses influence benthic communities by preying on crustaceans and mollusks, regulating populations that might otherwise overgraze algae. or disrupt coral recruitment. Shell middens, piles of discarded prey remnants outside dens, mark zones of concentrated predation, altering microhabitat composition and nutrient distribution. These localized impacts ripple outward, shaping community structure over time. The edited den is not an isolated event, but node in reef metabolism. There is also the paradox of distributed control. Unlike vertebrates with centralized brains commanding limbs, octopus arms possess significant neural autonomy. Each sucker can taste and feel, sampling chemical signatures directly from substrate. This decentralized intelligence allows simultaneous problem-solving across multiple contact points, accelerating adaptation within confined spaces. The octopus does not merely think about environment, it senses and computes through it, arms acting as exploratory extensions of cognition. The terrain is not external stage, but integrated sensory field. The final reversal reframes the octopus not as solitary trickster, but as participant in evolutionary dialogue with reef architecture. Crevices exist because corals grow and erode, because parrotfish grind limestone into sand, because storms fracture skeletons. The octopus exploits these processes, but also contributes to them through predation and den construction. Intelligence becomes ecological force, shaping patterns of survival in subtle ways. The magician edits stage, yet the stage edits magician in return. As the crab disappears into the living crack and the rock face resumes its static facade, the reef seems unchanged. Light flickers across stone, currents carry plankton, and small fish resume cautious patrol. Beneath that appearance lies a mind attuned to geometry, capable of reconfiguring shelter into snare and barrier into advantage. The trap is not buried or hidden. It is improved terrain, recalibrated by an animal that refuses to accept environment as fixed. In this labyrinth of coral and shadow, the octopus does not simply survive. It curates the maze. The sand ripples, but the ripple has eyes. A goby glides inches above the seabed, scanning for amphipods among grains sculpted by current. The pattern beneath it appears consistent. Pale ridges, darker troughs, scattered fragments of shell reflecting soft light. Then the pattern tightens. Chromatic bands sharpen where there were none before, and what seemed like random texture resolves into a pupil that dilates with intent. Two feeding tentacles uncoil with ballistic precision, propelled by muscular hydrostats that extend faster than the goby's neural reflex arc can process. The background inhales, and the goby is gone. The sand settles into neutrality again, as if it had never held a mind. The cuttlefish does not merely hide against its environment, it rewrites it. Its skin is a layered instrument of illusion, composed of chromatophores that expand and contract under direct neural control, iridophores that reflect polarized light, and leucophores that scatter ambient wavelengths to match surrounding luminance. These layers interact to generate patterns that mimic sand, coral rubble, seagrass shadows, or mottled reef floor with astonishing speed. Unlike static camouflage reliant on genetic coloration, the cuttlefish updates its display in fractions of a second, responding to visual input with dynamic output. The environment is not backdrop, but data stream. Vision drives this transformation. Cuttlefish possess large, camera-like eyes, with W-shaped pupils that enhance contrast detection in complex light fields. Their retinas are tuned to discern polarization patterns invisible to many other species, granting them a perceptual advantage in the shimmering coastal shallows where light fractures into angular noise. By analyzing edges, textures, and luminance gradients, the cuttlefish calculates which pattern class, uniform, mottle, or disruptive, will best dissolve its outline. The animal becomes algorithm embodied, translating visual context into dermal code. The first narrative reversal challenges the idea of camouflage as concealment alone. For the cuttlefish, disguise is also communication and control. When courting or confronting rivals, it can display high-contrast patterns on one side of its body while maintaining cryptic tones on the other, directing signals selectively. The same skin that erases presence can broadcast dominance. Camouflage here is multifunctional interface, not singular defensive trait. What appears as passive blending conceals a sophisticated signaling apparatus capable of rapid emotional expression and tactical misdirection. Locomotion integrates with illusion. Cuttlefish hover by undulating a thin fringe that encircles their mantle, allowing subtle adjustments without disturbing sediment. This method of movement minimizes hydrodynamic signature, preventing telltale bursts of turbulence that might alert prey. When attack is initiated, however, the calm collapses. Two specialized feeding tentacles, stored coiled within the arm crown, shoot forward under hydraulic pressure generated by muscular contraction. These tentacles, tipped with sucker pads, seize prey and retract with equal speed, delivering the victim to a beak concealed at the center. The transformation from stillness to strike is so abrupt that it appears discontinuous, as though the environment itself fractured. The cuttlefish's soft body, supported by an internal cuttlebone that regulates buoyancy, allows fine control of vertical position. By adjusting gas content within the porous cuttlebone, it can rise or sink without overt motion, maintaining precise alignment with substrate. This buoyancy control enhances its ability to hover just above sand or rubble, maintaining the illusion of static terrain. The environment is not only visual, but physical medium through which it calibrates position. The second narrative reversal emerges when the cuttlefish becomes prey. Dolphins, large fish, and sharks target cuttlefish, exploiting moments when disguise falters. In response, the cuttlefish deploys ink, not as mere cloud, but as structured decoy. Dense plumes form pseudomorphs, dark shapes approximating the cuttlefish's size, that linger as the animal jets away, using rapid mantle contractions. The ink also contains compounds that interfere with predator chemoreception, adding chemical confusion to visual deception. The live ambush screen becomes smoke and mirror, and the predator must reassess target identity. Intelligence extends survival beyond camouflage. Ecologically, cuttlefish occupy dynamic coastal zones where sand meets reef and vegetation interlaces with open patches. These habitats are characterized by shifting textures and fluctuating light, demanding constant recalibration of disguise. By preying on crustaceans and small fish, cuttlefish influence benthic population dynamics, shaping grazing pressure and nutrient cycling. Their predation can reduce densities of certain invertebrates, indirectly affecting algal growth and substrate stability. The ambush screen is therefore not isolated spectacle, but component of trophic regulation. Neurally, the cuttlefish is formidable. Its brain-to-body mass ratio rivals that of some vertebrates, supporting learning, memory, and problem-solving. Experiments reveal capacity for delayed gratification and adaptive foraging strategies, suggesting cognitive evaluation of future reward. In the wild, this intelligence manifests in flexible hunting approaches, stalking, flushing prey from cover, or positioning against current to intercept drifting organisms. The animal does not rely on single tactic. It improvises within environmental parameters. The flexibility of patterning destabilizes prey perception. In habitats where certain textures are assumed inert, the cuttlefish's ability to replicate those textures erodes reliability of visual cues. A fish navigating rubble cannot assume that mottled patch is lifeless. The background becomes suspect. This perceptual uncertainty increases cognitive load on prey, forcing constant vigilance that expends energy and reduces feeding efficiency. The trap is not solely physical capture, but psychological pressure exerted across ecosystem. Metabolic demands underpin this versatility. Maintaining neural processing and rapid chromatophore control requires energy, and cuttlefish exhibit high growth rates and relatively short lifespans. They must feed efficiently and often to sustain tissue and brain function. Failure to secure prey translates quickly into vulnerability. The shifting screen is expensive to operate. Intelligence and adaptability come at metabolic cost that must be paid through successful ambush. The final reversal reframes identity. The cuttlefish appears to dissolve into sand, yet it is neither sand nor fixed pattern. It is a living surface that can crystallize into face, arm, and intention without warning. For prey, the distinction between environment and organism collapses, The world is no longer reliable stage, but mutable interface where any texture might awaken. The trap is not hidden in shadow or buried under substrate. It is the substrate updated in real time. As currents smooth the seabed and the shimmer of light resumes its dance, the cuttlefish rests again as anonymous patch within larger mosaic. Its pupils narrow, its chromatophores relax, and its fin fringe hums with quiet equilibrium. The coastal zone remains dynamic, sand shifting grain by grain, coral fragments scattered across pale floor. Within that flux, a mind waits, capable of turning pattern into predator at will. The difference between harmless texture and living hunter is measured in milliseconds, and in that sliver of time, the background itself can become a face. The current does not feel dangerous. It slides through the seagrass meadow in soft pulses, bending blades in synchronized arcs, carrying with it a snowfall of plankton, larval crustaceans, and drifting copepods that rise and fall with each tidal breath. Within that gentle choreography, a translucent amphipod tumbles sideways, legs paddling against nothing substantial, its compound eyes scanning for predators that lunge or flash or cast shadows. There is no flash here, no shadow worth noting, only a thin strand of grass swaying with identical rhythm to its neighbors. The strand has a face. The strand has eyes that pivot independently, tracking the amphipod's erratic path with neural precision, refined over millions of years of ambush in slow water. Then the meadow inhales, and the amphipod disappears in a silent implosion of suction so subtle that the surrounding blades do not tremble. The seahorse does not hunt by speed or terror. It hunts by architecture. Its body is upright, armored in bony plates rather than scales, and coiled at the tail into a prehensile anchor that grips seagrass stems or coral branches with muscular certainty. This posture appears fragile, almost ornamental, but it is biomechanical strategy disguised as delicacy. By orienting vertically, the seahorse reduces its lateral profile against the dense vertical lines of vegetation, dissolving into the geometry of the habitat. Camouflage here is not coloration alone, although chromatophores do allow subtle shifts in hue to match green, brown, or yellow tones. It is alignment. The seahorse becomes one more blade in a forest that moves in unison. Hydrodynamics dictates its feeding corridor. Coastal shallows and estuarine beds are defined by microcurrents shaped by tidal oscillation, temperature gradients, and the roughness of substrate. Tiny invertebrates drift not randomly, but along predictable flow paths where turbulence meets laminar slip. The seahorse chooses position within these lanes, selecting attachment points that maximize prey encounter rate while minimizing exposure to larger predators. Its decision is not conscious planning in human terms, yet it reflects evolutionary tuning of behavior to fluid physics. The meadow becomes conveyor belt, and the seahorse becomes gatekeeper. Its feeding apparatus is a study in precision engineering. The elongated tubular snout functions as a rapid suction pump driven by expansion of the buccal cavity. When prey crosses a threshold distance measured in mere centimeters, the seahorse rotates its head upward with a specialized pivot joint at the neck, known as a 4-bar linkage system, a rare skeletal configuration among fishes. This mechanism amplifies angular acceleration, allowing the head to snap upward while the body remains anchored. The sudden expansion of the mouth cavity generates negative pressure, pulling water and prey inward in less time than the prey's escape neurons can trigger a tail flick. The strike is not chase, but capture through physics, converting fluid dynamics into lethal advantage. There is a metabolic constraint underlying this stillness. Seahorses lack a true stomach. Food passes rapidly through a simplified digestive tract, limiting nutrient extraction and forcing frequent feeding. Their calm posture is not laziness, but necessity. They must intercept hundreds of tiny prey items daily to sustain tissue, growth, and reproduction. The feeding lane is therefore not occasional convenience, but continuous survival channel. If current shifts or vegetation thins, energy intake falters quickly, and the cost of patience becomes starvation. The trap is delicate because it must be constantly maintained through position choice and micro-adjustment. The first narrative reversal unfolds when we consider vulnerability. Anchored and slow, seahorses appear easy targets for predatory fish. Their swimming ability is limited to gentle propulsion using a rapidly beating dorsal fin that vibrates like a humming membrane, and they cannot flee across long distances. Yet this apparent weakness is transformed into defense through structural and behavioral specialization. Their bony exoskeleton deters some predators by reducing palatability and flexibility. Their cryptic coloration and tendency to sway mimicry reduce detection probability. By staying embedded in vegetation, they avoid entering open water where pursuit speed matters. What seems like helplessness is ecological specialization optimized for narrow niche. Reproduction intensifies the reversal. In a biological twist that unsettles conventional expectations, it is the male seahorse that becomes pregnant. The female deposits eggs into a brood pouch on the male's abdomen, where vascularized tissue regulates salinity, oxygenation, and nutrient transfer. This paternal investment changes social dynamics. Males anchored within feeding lanes carry developing embryos, while still needing to maintain energy intake. The brood pouch functions almost like a temporary placenta, modulating ionic composition to match external seawater and internal embryonic needs. The meadow becomes nursery as well as hunting ground, and the anchored predator becomes incubator of life. This reproductive strategy alters population resilience. By distributing parental care between sexes in atypical fashion, seahorses may stabilize embryo survival in environments where egg predation is high. However, it also constrains mobility further. A brooding male cannot risk relocation lightly, tying reproductive success to habitat stability. Seagrass beds therefore become critical infrastructure, not simply backdrop. When coastal development, pollution, or rising temperature degrade these habitats, the feeding corridor collapses. Prey distribution shifts, and the micro-trap loses alignment. The ecological implications ripple outward. Seahorses serve as indicators of seagrass health, and their decline signals broader trophic imbalance. The second narrative reversal emerges at ecosystem scale. What appears as solitary decorative fish is participant in complex food web interactions. By consuming large numbers of small crustaceans, seahorses influence planktonic community composition and potentially affect grazing pressure on microalgae. In dense populations, their predation may alter distribution of certain copepods or amphipods, subtly shifting nutrient cycling within meadow. At the same time, they are prey for larger fish and crabs, linking primary consumers to higher trophic levels. The micro-trap is embedded in macro dynamics, its quiet strikes contributing to energy transfer across ecosystem. Sensory adaptation deepens appreciation. Seahorses possess independently moving eyes, enabling them to scan for prey and predators simultaneously. This binocular independence increases visual field without body motion, preserving camouflage while enhancing awareness. Neural processing integrates these dual streams, allowing calculation of prey trajectory against background motion of grass. The environment is dynamic noise. The seahorse extracts signal from that noise through evolved perception tuned to flicker and drift. Each successful suction strike reflects millisecond coordination between sensory input and mechanical output. Temperature and salinity fluctuations in shallow waters impose physiological stress. Seahorses must tolerate variable oxygen levels and occasional turbidity. Their gills and circulatory systems adjust to maintain homeostasis under such conditions, but extremes can push limits. Climate-driven changes that intensify heat waves or alter current regimes threaten the delicate balance of feeding lanes. The micro-trap depends on predictable flow patterns. When currents accelerate beyond typical range, prey may bypass anchored hunters, and when stagnation reduces plankton availability, starvation risk rises. The still hunter is hostage to moving water. Yet within this fragility lies resilience shaped by evolutionary time. Fossil records suggest that ancestral cyngnathids adapted gradually to coastal vegetated habitats, refining prehensile tails and upright posture as selective advantages in structurally complex environments. The seahorse's form is culmination of iterative adjustments to hydrodynamic and ecological pressures. What appears whimsical silhouette is record of environmental negotiations spanning millions of years. Its architecture is biography written in bone. The final reversal reframes perception of drama. In open ocean narratives, danger is spectacle, breaching predators, explosive chases, violent collisions. Here, the trap is quiet to the point of invisibility. The seahorse does not rupture surface or scatter schools in panic. It engineers probability. By anchoring at intersection of current and cover, it increases likelihood that drifting life will cross a narrow zone where suction can convert motion into nourishment. The violence is microscopic and continuous, measured not in splashes, but in thousands of tiny disappearances that sustain fragile body. As tide shifts and meadow leans in new direction, the seahorse tightens its grip and adjusts orientation, recalibrating to fresh angle of flow. Planktonic snow resumes its descent through green lattice, and another amphipod drifts unwittingly along invisible corridor. The blades sway, the light refracts, and the difference between plant and predator blurs again. Not every ocean trap roars or lunges. Some are woven from patience, physics, and a tail wrapped quietly around a stem, turning gentle water into narrow lane of irreversible fate. The snail shell fractures before the water can register the sound. A hairline crack blooms across calcium carbonate that took months to build, and in less time than a neuron can complete a reflex arc, the crack becomes collapse. The predator does not pursue, it detonates. From the dark aperture of a coral rubble burrow, a club-shaped appendage arcs outward with such acceleration that the surrounding seawater flashes into vapor cavities, cavitation bubbles that implode with micro-explosions, rivaling the force of the initial blow. The doorway looks small, inconsequential, an irregular hole between broken coral branches. It is a checkpoint. It is a weaponized threshold. The mantis shrimp does not need open water to dominate. It needs geometry. By anchoring itself within a burrow excavated under rock or inside consolidated rubble, it reduces the world to a narrow field of approach where reaction time, not body size, determines outcome. Any crab probing for shelter, any snail grazing along the substrate, any rival stomatopod assessing territorial boundaries must pass through a constricted corridor of detection and response. In this confined arena, the mantis shrimp's neuromuscular system becomes decisive. Its raptorial appendages, evolved into either smashing clubs or spear-like blades depending on species, are powered by a latch and spring mechanism that stores elastic energy in a saddle-shaped structure composed of mineralized cuticle. When released, that stored energy converts into rotational acceleration, so extreme that the appendage can reach velocities capable of shattering shells and even aquarium glass. The environment is not backdrop, it is amplification chamber. The strike is not mere impact, it is hydrodynamic event. As the club accelerates, it displaces water so rapidly that pressure drops below vaporization threshold, forming cavitation bubbles that collapse with additional shock waves. These implosions can stun or damage prey, even if the initial contact glances. The mantis shrimp wields physics beyond muscle alone, leveraging fluid dynamics as secondary weapon. The doorway, therefore, is a focal point where mechanical energy and fluid instability converge. Any organism crossing that boundary enters a microenvironment primed for explosive release. Vision completes the system. Mantis shrimp possess one of the most complex visual apparatuses in the animal kingdom, with compound eyes mounted on independently mobile stalks, capable of trinocular vision in each eye. Their retinas are divided into multiple bands of photoreceptors sensitive to a broad spectrum of wavelengths, including polarized light. This capacity allows detection of subtle movements and polarization cues reflecting off scales or shells, even within cluttered reef substrate. The burrow entrance becomes surveillance station, and the mantis shrimp reads the optical signature of approaching life with extraordinary acuity. The checkpoint is guarded by perception, tuned to nuances invisible to many prey species. The first narrative reversal emerges when we consider scale. In open water, a small crustacean would be outmatched by larger fish or cephalopods. Within its burrow domain, however, the mantis shrimp neutralizes size advantage by constraining engagement distance. Large predators cannot easily insert their jaws into narrow crevices without exposing vulnerable tissue,
and the mantis shrimp's strike can deter even those many times its mass. What appears diminutive becomes sovereign within confined architecture. Territoriality is not defended by bulk, but by speed and geometry. Territory is more than refuge, it is extension of body. Mantis shrimp meticulously maintain their burrows, clearing debris, adjusting entrance angle, and reinforcing walls with sand and rubble. This maintenance ensures optimal field of fire and escape routes through secondary tunnels. The burrow functions as both fortress and springboard, a place where elastic energy is primed in relative safety before release. In ecological terms, such behavior exemplifies niche construction, whereby organism modifies environment to enhance survival prospects. The mantis shrimp edits substrate into kinetic landscape, where its adaptations yield maximal effect. Metabolic demands drive this ferocity. Rapid strikes and active burrow maintenance require significant energy, and mantis shrimp feed primarily on hard-shelled mollusks, crabs or fish depending on species specialization. Smashing species break through exoskeletons that deter other predators, accessing nutrient-rich tissues inaccessible to slower hunters. Spearing species impale soft-bodied prey with bladed appendages lined with barbs. In both cases, the burrow's proximity to foraging grounds allows efficient energy expenditure. By minimizing pursuit distance and maximizing capture probability within controlled space, the mantis shrimp aligns energetic output with intake in a tight budget. The second narrative reversal complicates perception of brutality. Though armed with devastating force, mantis shrimp engage in elaborate ritualized displays when encountering rivals of similar size. They raise their bodies, spread appendages, and perform merrill spreads, visual presentations of colored patches on the raptorial limb that signal capability without immediate violence. Such displays reduce frequency of lethal combat, conserving energy and avoiding injury that could compromise survival. The same appendage that can fracture shell can also communicate restraint. Aggression is modulated by assessment, not blind impulse. Their sensory world is layered beyond human intuition. The ability to detect circularly polarized light, rare among animals, may enhance discrimination of transparent prey or signal quality among conspecifics. Polarization sensitivity also penetrates glare in shallow water, sharpening contrast where sunlight fractures into chaotic reflections. From within the burrow's shadow, the mantis shrimp interprets this complex lightscape with neural circuits capable of rapid processing. Reaction time is measured in milliseconds, and synaptic transmission must be correspondingly swift. The checkpoint is guarded not only by muscle, but by a brain tuned to instantaneous decision-making. Predators do exist. Larger fish, octopuses, and even other mantis shrimp can threaten burrow residents. Here, the doorway becomes vulnerability as well as defense. A predator that blocks escape routes or collapses the burrow roof can trap the mantis shrimp within its own fortress. Thus, burrow placement matters. Selecting stable substrate and multiple exits reduces risk. The architectural intelligence extends to choosing sites with limited overhead exposure, ensuring that the narrow entrance remains defensible rather than trap. What seems impregnable can invert into confinement if misjudged. At ecosystem scale, mantis shrimp influence benthic community structure. By preying on mollusks and crustaceans, they affect shell density and invertebrate populations, potentially altering competition dynamics among grazers. Their burrowing activity aerates sediment, contributing to nutrient cycling and oxygen penetration within substrate layers. The violent strike at doorway reverberates through trophic webs, shaping distribution of species across reef flats and sandy bottoms. The checkpoint is node within larger network of ecological feedback. Climate shifts and habitat degradation introduce new pressures. Coral rubble fields may diminish as reef frameworks erode under warming and acidification, reducing availability of suitable burrow sites. Increased turbidity from coastal development can alter prey distribution and visual clarity, challenging the mantis shrimp's reliance on optical detection. Ocean acidification also affects shell integrity of prey species, potentially changing energetic cost-benefit of smashing strategy. If shells weaken, strike force may exceed necessary threshold, but prey availability may fluctuate unpredictably. The balance between mechanical capacity and ecological opportunity evolves under environmental stress. The final reversal reframes violence as specialization rather than excess. The mantis shrimp's extraordinary strike is not gratuitous brutality, but solution to evolutionary puzzle posed by armored prey and confined habitats. In a world where many predators cannot penetrate hard exoskeletons without prolonged effort, the mantis shrimp resolves resistance in single explosive event, reducing struggle time and exposure to its own predators. The doorway is not only hazard for others, but efficiency engine for itself. By converting stored elastic energy into decisive action, it transforms tiny territory into high-risk zone for any organism that miscalculates proximity. As dusk filters through reef and shadows lengthen across rubble, the burrow entrance remains unremarkable, a small dark circle among broken coral and sand. Copepods drift, hermit crabs scuttle, and small fish forage with cautious curiosity. The mantis shrimp waits just inside, appendages folded, elastic structures primed beneath armored carapace. In this corner of the ocean, size hierarchy dissolves at threshold. The next probe too close, the next careless graze across invisible boundary, will trigger a release of stored tension that collapses shell and certainty alike. The doorway does not announce its danger. It simply holds it, coiled and patient, until motion crosses the line. The reef does not explode with panic. It tightens. A cloud of juvenile damselfish flickers above branching coral, their bodies flashing silver as they graze on drifting plankton. There is no sudden rush, no torpedoing silhouette slicing through the school. Instead, a striped figure glides forward with the unhurried certainty of something that has never needed to chase. Its pectoral fins unfurl like ornate fans, translucent membranes edged with venomous spines that radiate outward in a widening arc. The water in front of it seems to shrink. The damselfish pivot, trying to slip sideways, but the sideways has disappeared. The coral wall is behind them. The lionfish is in front of them. The distance between those two realities is closing. The lionfish does not hunt with speed, it hunts with geometry. Its elongated dorsal spines and broad pectoral fins are not merely decorative warnings of venom. They are tools of spatial manipulation. By spreading its fins into a semicircular barrier, the lionfish alters the hydrodynamic landscape immediately around it, creating a visual and physical boundary that compresses prey into tighter formations. Small fish, wired to avoid sudden lunges, misinterpret the lionfish's slow advance as non-threatening drift, even as their available escape vectors narrow to a vanishing point. The reef wall becomes part of the predator's body, a hard surface against which prey are gently but inexorably pressed. The lionfish's strike is a rapid inhalation rather than a chase. As it closes the final gap, it flares its buccal cavity, expanding the mouth so suddenly that water and prey are drawn inward through suction. The movement is precise, controlled, and often invisible to anything beyond immediate vicinity. By the time the damselfish recognize the danger, the pressure differential has already collapsed their escape into ingestion. The lionfish does not need to outrun, it only needs to reduce options until the physics of water flow finishes the job. This method relies on reef structure. Coral heads, overhangs, and crevices create dead ends that prey often use as shelter. Lionfish exploit these microhabitats by positioning themselves between open water and refuge, effectively transforming safe space into trap. Their vertical orientation among sponges and branching coral allows them to approach from angles that minimize silhouette detection. Unlike ambush predators that vanish into substrate, lionfish remain visible yet misinterpreted, their ornate fins and bold stripes blending into the visual complexity of reef texture. Pattern recognition in prey species fails under sensory overload, and the lionfish capitalizes on that confusion. Venom adds another layer of control. The dorsal, pelvic, and anal spines are equipped with venom glands capable of delivering painful injections to would-be predators. This defense reduces harassment and allows lionfish to hunt boldly in daylight when many reef predators might otherwise dominate. The apparent fragility of extended spines becomes protective armor. Predators that attempt to swallow a lionfish risk envenomation, and many learn to avoid them after initial encounter. The hunter is insulated from retaliation by biochemical deterrent, freeing it to refine spatial manipulation tactics without constant fear. The first narrative reversal emerges when we examine the lionfish's status beyond its native Indo-Pacific range. In regions such as the Western Atlantic and Caribbean, lionfish have become invasive, introduced through aquarium releases, and now proliferating across reefs unaccustomed to their tactics. Native prey species in these ecosystems lack evolutionary history with lionfish and often fail to recognize them as predators. The slow advance and fin display do not trigger avoidance behaviors calibrated for faster, more familiar threats. As a result, lionfish densities have surged, and their predation pressure has altered community structure, reducing recruitment of native fish and potentially affecting reef resilience. The elegant trap becomes ecological upheaval. Their reproductive strategy amplifies this impact. Lionfish are highly fecund, releasing gelatinous egg masses that drift with currents, dispersing larvae across vast distances. This planktonic phase allows colonization of new reefs far from original populations. Once settled, juveniles mature quickly and integrate into reef food webs as efficient predators. The calm hunter multiplies silently, each individual capable of consuming significant biomass of small fish and crustaceans. In invaded ecosystems, this predation can reduce herbivorous fish populations that normally graze algae, indirectly promoting algal overgrowth on coral surfaces. A trophic cascade begins with slow pressure at microscale and expands to reef-wide consequences. The second narrative reversal shifts focus to the lionfish itself as prey. Although venom deters many predators, some species have adapted to consume lionfish safely. Groupers and certain sharks have been observed preying upon them, sometimes after learning to flip or disable them to avoid spines. Human divers, recognizing ecological threat in invaded regions, have initiated targeted removals, turning lionfish into culinary commodity. The once unchallenged invader becomes hunted, and its ornate fins that once blocked escape lanes become signals for spearfishers seeking control. The trap built in motion encounters counterpressure from larger ecological and human systems. Lionfish sensory biology underpins their method. Their large eyes enhance low-light vision, allowing crepuscular and nocturnal hunting when prey visibility is reduced. Lateral line systems detect subtle water movements, informing the predator of prey shifts even when visual cues are minimal. Combined with deliberate fin positioning, this sensory integration allows lionfish to anticipate escape attempts and adjust orientation accordingly. The reef corridor becomes dynamic chessboard, each movement countered by incremental repositioning of spines and membranes. Energy efficiency characterizes their hunting style. Instead of high-speed pursuits that demand rapid bursts of muscular exertion, lionfish conserve energy through slow approach and precise suction strikes. This strategy aligns with metabolic economy in complex habitats where constant chases would risk collision with coral and injury. By turning structure into ally, lionfish reduce the need for stamina-based dominance. The trap is economical, trading spectacle for steady intake. Coral reef degradation introduces new complexities. As bleaching events and storm damage convert living coral into rubble fields, structural complexity can decline, potentially altering effectiveness of lionfish herding tactics. Yet rubble still offers corners and dead ends, and lionfish adapt by exploiting whatever geometry remains. In invaded regions where reefs are already stressed by warming and pollution, lionfish predation compounds existing vulnerabilities. The slow pressure applied at small scale accumulates across seasons, reshaping species composition. Behaviorally, lionfish sometimes cooperate loosely by hunting in proximity to other individuals, potentially enhancing herding effect. While not true coordinated pack hunting, overlapping territories can create zones where prey are funneled between multiple predators. The reef geometry thus becomes multi-layered trap, with fins and coral forming interlocking barriers. The spectacle remains understated, yet mortality rises for anything caught in converging corridors. The final reversal reframes perception of ornamentation. What appears flamboyant and conspicuous, striped body, flowing fins, serves as functional apparatus for spatial dominance and chemical defense. The lionfish does not hide. It persuades prey into complacency before compressing their world into a narrowing funnel.
Its beauty is mechanical strategy disguised as decoration. In ecosystems where its presence is natural, it occupies balanced niche among other predators, contributing to dynamic equilibrium. In ecosystems where it is alien, that same strategy destabilizes networks built without anticipation of such slow-moving blockade. As currents shift and reef shadows lengthen, another group of juveniles drifts toward a coral head, unaware that the open water behind them is quietly sealing. The lionfish advances without hurry, fins extended in silent proclamation of control. There is no explosive chase, no dramatic breach. There is only the steady subtraction of exits until motion itself becomes error. In this part of the ocean, danger does not announce itself with speed. It arrives as narrowing space, as reef-turned accomplice, as gentle pressure that leaves prey nowhere left to turn. A parrotfish lowers its beak toward a carpet of algae and hesitates. The reef slope is quiet, sunlit, seemingly benign, yet the rock beneath the green film bristles with motionless spears. Hundreds of spines radiate outward from spherical bodies, wedged into crevices and scattered across the limestone like living caltrops. The parrotfish tests the surface with a cautious nudge, then withdraws abruptly as a spine flexes and snaps back with elastic resistance. What appears to be inert rubble is in fact a field of conditional pain, a biomechanical deterrent that reshapes every decision taken above it. Sea urchins do not lunge, chase, or camouflage. They fortify. Their bodies are encased in rigid tests composed of interlocking calcium carbonate plates, forming a near-perfect sphere that distributes mechanical stress evenly across its surface. From this armored core emerge spines articulated by ball and socket joints, capable of subtle, coordinated movements driven by muscular control. These spines are not static fences. They pivot, converge, and respond to tactile stimuli, transforming the seafloor into reactive hazard. Any predator attempting to grip or bite must navigate a lattice of sharp projections that can pierce soft tissue and resist removal. In certain species, the deterrence escalates through venom. The spines of some tropical urchins are hollow and connected to venom glands, delivering toxins that cause intense pain and inflammation upon penetration. Others possess pedicellaria, tiny pincer-like appendages between the spines that snap shut on contact, injecting venom or mechanically gripping skin. These microstructures function as secondary defense, ensuring that even if a spine is avoided, another mechanism stands ready to impose cost. The bottom is not simply covered, it is defended at multiple scales. The ecological effect of this defensive architecture extends far beyond individual survival. When urchin densities increase, whether through reduction of predators or environmental shifts, the substrate transforms into restricted ground. Grazing fish that rely on close contact with rock surfaces must adjust feeding strategies to avoid injury. Sea turtles searching for resting spots may detour around clusters of spines. Even currents interact differently with dense aggregations, as the spines alter boundary layer flow near the substrate, influencing sediment deposition and nutrient exchange. The reef floor becomes textured not only visually but behaviorally. The first narrative reversal challenges the assumption of passivity. Sea urchins, often perceived as slow and exposed, can exert disproportionate influence over ecosystem structure. By grazing algae with specialized mouthparts known as Aristotle's lantern, a complex arrangement of 5 calcareous teeth capable of scraping rock, they control algal growth and influence coral recruitment. In balanced systems, moderate urchin grazing prevents algae from smothering corals, maintaining reef resilience. Yet when predator populations decline, urchin numbers can surge, leading to overgrazing that strips surfaces bare and creates so-called urchin barrens. The defensive sphere becomes ecological engineer, capable of shifting entire reef states. Their feeding activity involves more than simple scraping. As urchins rasp algae from rock, they also erode limestone substrate, contributing to bioerosion processes that shape reef morphology over time. This erosion releases calcium carbonate particles into surrounding waters, subtly altering sediment composition and influencing microhabitats for invertebrates. The spiny fortress that deters predators simultaneously participates in geological transformation, linking individual feeding to long-term structural evolution of the reef. Predator-prey dynamics introduce further complexity. Many fish species, including triggerfish and some wrasses, have evolved strategies to overcome urchin defenses. Triggerfish may flip urchins onto their backs, exposing the vulnerable oral surface where spines are shorter and movement less coordinated. Sea otters in temperate kelp forests crack urchins open with stones, bypassing spines entirely. Here emerges the second narrative reversal. The field of consequences that once dictated avoidance can itself become target when predators adapt. In kelp ecosystems, sea otter predation on urchins reduces grazing pressure on kelp, allowing forests to flourish. Remove the otters and urchins proliferate, decimating kelp beds and creating barren landscapes. The spines that once shaped behavior become drivers of trophic cascades. Sea urchin reproduction amplifies their ecological reach. Many species release eggs and sperm into the water column in synchronized spawning events, relying on currents to disperse larvae. These planktonic larvae can travel significant distances before settling, colonizing new substrates. Settlement patterns are influenced by chemical cues from algae and biofilms, meaning that urchin distribution reflects interplay between larval behavior and environmental signals. When conditions favor high recruitment, the seafloor can rapidly transition from sparsely occupied to densely armored. Physiological adaptations support this defensive lifestyle. Tube feet extending from ambulacral grooves allow slow but steady movement, adhering to rock through suction and muscular contraction. These tube feet also assist in righting the urchin if overturned, a vulnerable state that exposes softer tissues. Coordination between spines and tube feet enables subtle repositioning in response to light, current, or predator approach. Though lacking centralized brain, urchins possess a nerve ring and radial nerves that integrate sensory input across their body, allowing distributed decision-making across a decentralized nervous system. The fortress responds as unit without need for head. Environmental stresses influence urchin populations and defensive efficacy. Ocean acidification can weaken calcium carbonate structures, potentially compromising spine integrity and test strength. However, some species exhibit physiological plasticity, adjusting calcification rates under changing pH conditions. Temperature shifts can alter reproductive timing and larval survival, affecting density and distribution. In regions where overfishing reduces predators, urchins may thrive despite environmental pressures, underscoring the interplay between human activity and natural dynamics. The behavioral consequences of urchin fields ripple outward. Fish that might otherwise graze evenly across reef surfaces cluster in safer zones, concentrating feeding pressure and potentially altering algal composition. Invertebrates seeking shelter must navigate spine arrays, sometimes coexisting within urchin shadows to gain protection from larger predators. Certain small fish and shrimp species use urchin spines as refuge, weaving among them with precision. Thus, the painful obstacle course doubles as sanctuary for some, revealing a nuanced layer within apparent hostility. The trap that deters one species shelters another. In temperate kelp forests, dense urchin populations can transform lush underwater forests into stark barrens where only encrusting coralline algae persist. The removal of kelp reduces habitat complexity and carbon sequestration capacity, altering energy flow through the ecosystem. When sea otters or other predators restore balance by reducing urchin numbers, kelp can rebound, re-establishing canopy structure and associated biodiversity. The spines, though static in appearance, sit at nexus of dynamic feedback loops involving herbivory, predation, and habitat engineering. The final reversal reframes perception of aggression. Sea urchins do not pursue, yet they impose constraints that shape movement patterns of entire communities. Their defense is spatial taxation. Any organism crossing their domain pays in caution, injury risk, or detour. Over time, such taxation influences distribution of species, patterns of grazing, and even success of coral recruitment. The seafloor ceases to be neutral plane and becomes mosaic of safe and restricted zones determined by density of spines. As currents sweep across reef and light filters down in shifting bands, the urchin field remains outwardly still. Spines sway gently with water motion, casting thin shadows across rock. A parrotfish circles, selecting a less defended patch to graze. A small shrimp slips between spines, sheltered within labyrinth of points. The sphere of calcium carbonate and keratin-like spines exerts influence without movement, a quiet dominion that alters behavior through anticipation of pain. Not all traps snap shut. Some endure as landscapes of consequence, reshaping the choices of every fin, foot, and mouth that dares approach. The sand exhales before it moves. A faint plume rises, as if the seabed itself has sighed, and then the outline appears. Two eyes lifting through a veil of grains, spiracles pulsing like discrete valves that ventilate a hidden body. A juvenile mullet, grazing without urgency along the shallows, darts across this patch of bottom as though it were empty ground. In the next instant, the ground folds. A disc-shaped form surges upward, pectoral fins sweeping in a coordinated undulation that displaces water and sediment simultaneously, and the mullet vanishes beneath a curtain of suspended sand. What looked like neutral terrain was an organism poised at the boundary between concealment and propulsion, and the boundary has just been breached. The stingray does not rely on absence, it relies on misinterpretation. By partially burying itself beneath substrate, using oscillations of its broad fins to loft sediment over its dorsal surface, the ray reduces its visible profile to subtle contours that resemble irregularities in sand. Its dorsum often matches the coloration of local sediment, from pale beige to mottled brown, rendering it visually continuous with the seabed. Only the protruding eyes and spiracles betray its presence, and even these structures are positioned on top of the body so that respiration and surveillance can continue while the rest remains concealed. The ocean floor is not a background for the stingray. It is an extension of its integument. Respiration itself reveals evolutionary engineering aligned with benthic life. Unlike many fishes that draw water through their mouths and expel it over gills, stingrays can intake water through spiracles positioned dorsally, channeling it to gill slits located ventrally. This arrangement allows them to breathe while resting or hunting with mouths pressed against substrate, avoiding ingestion of sediment that would clog gill filaments. The physiological adaptation supports the behavioral strategy of burial, maintaining oxygen flow even when the mouth is engaged in feeding or concealed under sand. The floor becomes breathable shelter rather than suffocating cover. When the ray hunts, it does not merely wait for prey to pass. It actively reshapes the bottom. Using rhythmic undulations of its pectoral fins, it can create localized depressions in sand, exposing buried invertebrates such as clams, worms, and crustaceans. Electroreceptors embedded around its mouth detect weak bioelectric fields emitted by muscular contractions of hidden prey. Even when visual cues are obscured by sediment, the ray senses neural activity beneath the surface, translating electrical whispers into targeted excavation. The seabed becomes acoustic and electrical landscape, readable in ways invisible to other inhabitants. The first narrative reversal complicates the perception of the ray as passive ambusher. In some coastal systems, rays form aggregations that traverse tidal flats, collectively disturbing sediment as they forage. This disturbance can influence distribution of invertebrates and alter nutrient cycling within benthic layers. By turning over sand and redistributing organic matter, rays participate in bioturbation processes that affect microbial communities and oxygen penetration into substrate. The apparent solitary hunter reveals itself as agent of ecosystem engineering, reshaping the very ground it uses for concealment. Defense introduces the second layer of consequence. The tail spine, often serrated and coated in venom-producing tissue, is positioned along a muscular tail that can whip upward in defensive arc. The stingray does not wield this spine as offensive spear in routine hunting. It is deployed primarily when the animal is startled or restrained. Yet its presence transforms human and animal interactions alike. A foot descending onto buried ray triggers rapid reflexive strike, embedding spine into soft tissue and injecting venom that causes intense pain and inflammation. The sandy shallows, often perceived as safe recreational space, conceal this conditional hazard. The floor retaliates when misjudged. Predator-prey dynamics further nuance the picture. Large sharks and marine mammals prey upon stingrays, sometimes flipping them to access softer ventral surfaces and avoid tailspine. In regions where shark populations decline, ray numbers may increase, intensifying predation pressure on benthic invertebrates and potentially altering community structure. Thus, the defensive weapon that deters many attackers does not render rays invulnerable. It simply modifies which predators can successfully exploit them. The spine shapes trophic interactions by filtering who can engage and who must retreat. The ray's flattened body plan optimizes hydrodynamic efficiency near substrate. By distributing mass across wide disc and using pectoral fins as primary locomotory surfaces, rays can glide with minimal energy expenditure, conserving metabolic resources in environments where prey density may fluctuate. This energy economy aligns with life in dynamic coastal zones where tides expose and submerge feeding grounds in rhythmic cycles. The stingray's metabolism and morphology are synchronized with temporal rhythms of shoreline ecosystems, allowing exploitation of windows when prey are most accessible. Environmental change introduces new dimensions. Coastal development increases sedimentation and alters water clarity, affecting ray hunting efficiency and prey distribution. Warmer waters may shift ranges of both rays and their predators, recalibrating interactions across regions. Ocean acidification impacts calcifying organisms such as clams and crustaceans, potentially modifying prey availability and shell integrity. The stingray's strategy, melding with sand, sensing electricity, striking with sudden propulsion, must adapt to substrates and communities in flux. The second narrative reversal emerges when considering human perception. Rays are often feared for their stings, yet incidents typically arise from accidental contact rather than predatory intent. Their defensive reflex is triggered by compression or surprise, not pursuit. The same organism that appears dangerous underfoot is, in most contexts, a cautious feeder more interested in invertebrates than confrontation. Education campaigns encourage shuffling feet in shallow water to alert buried rays, reducing surprise encounters. The trap is conditional, activated by ignorance rather than malice. What seems aggression is protective response rooted in vulnerability. Sensory adaptations extend beyond electroreception. Rays possess lateral line systems attuned to water movement, allowing detection of approaching predators or prey. Their eyes, though limited in field when buried, can track overhead silhouettes that signal danger. Coordination between sensory input and muscular output must be rapid. A delay could result in predation by sharks or entanglement in nets. Neural circuits integrate mechanical, electrical, and visual information into split-second decisions, remain buried, burst upward, or flee with undulating propulsion. In certain species, seasonal migrations occur between deeper offshore habitats and shallow coastal flats. These movements are driven by reproductive cycles, temperature gradients, and prey availability. Mating behavior involves complex interactions where males grasp females with modified pelvic fins, and females invest significant energy into gestation of live young, nourished through yolk and uterine secretions. The life history strategy links benthic camouflage with pelagic dispersal phases, weaving the ray into broader oceanic narratives beyond the sand patch where it rests. The final reversal reframes the concept of ground. For many marine animals, the seafloor is substrate, stable, inert, predictable. For the stingray, it is interface, shield, and weapon platform. By integrating body with sediment, the ray erases boundary between organism and environment, turning geology into biology. The ocean becomes most deceptive when the surface beneath our gaze is animate, capable of sensing, responding, and retaliating. As the tide recedes and sunlight fractures across shallow flats, the sand resumes its quiet shimmer. Only faint impressions hint at the bodies concealed below. A small crab scuttles past one such depression, unaware of the electric fields radiating from its own muscles. A sudden surge, a bloom of sand, and the crab is gone. The surface settles again, indifferent and unchanged. In this final movement of the episode, the ocean completes its transformation from backdrop to participant. The ground itself holds intention, and every step or fin stroke across it is a negotiation with life hidden just beneath the grains.